So this is how transcription and translation works. First, we start off with a gene. If you look closely into a gene, you get DNA. DNA is a double helix produced of 4 nitrogenous bases with a sugar phosphate backbone. A polymerase comes in and turns that DNA into RNA, and
then the process starts. So first we have transcription, which is the copying. So we start out with a piece of DNA, which has a 5' to 3' end and a 3' to 5' end. And DNA has a Tata box, which is pretty universal. And the Tata box, along with the transcription factors, start the initiation. And then during elongation, the RNA polymerase goes along copying the nitrogenous bases. And for RNA, it changes thymine to uracil. And so that's the RNA that's being created, and as it's going along, it's unzipping, copying, and basically pasting it below in a new strand of mRNA. And then it reaches the termination signal, and it stops, and then we have a completed strand of mRNA. And then to the mRNA, a 5' cap and a poly A tail are added, and these help the mRNA to be able to leave the nucleus, and it also helps the RNA attach to what it needs to later, and it helps protect it from enzymes that are just floating around. And before it gets to leave, it has to go through an editing process, and in this, exons are things that get added in, and these are good things that it needs later. And then introns are things that are bad, so they get taken out so it can do what it needs to do in translation.
During translation, the mRNA is going to come in and attach to the ribosome, and then the tRNA is going to come in, and the anticodons are going to attach to the start codons right here.
The tRNA then goes to the A site and brings in an amino acid that correlates with the codons. It brings it to the E site and then goes back to the A site and repeats the process, eventually making a polypeptide chain.
once the chain is created and all the amino acids are bonded, a protein is made and it goes off into the cell to do its protein duties.
Yeah.